Hello and welcome Stage 2 SMAF chemistry students. Today we're going to be looking at a type of analytical technique called chromatography. This is the third of the types of analytical techniques that we will be studying in this course. You may remember that we have so far done volumetric analysis, the first type of analytical technique we looked at, which essentially uses chemical reactions to work out the quantities of certain substances within a solution or any other substance. That is known as a quantitative type of analysis. We can work out the amounts of a particular substance within a solution or any other type of mixture. The second being an atomic absorption spectroscopy or atomic spectroscopy that was the second type of analytical technique that we have looked at, and that is around using the unique nature of elements and their ions in terms of the spectra of light that they emit to identify substances within a sample of anything. And this can be a qualitative and quantitative, because we can actually work out what is within a substance or a sample that we might get, and we can also work out the amount using atomic absorption spectroscopy. So that is what we call both a qualitative and quantitative type of analysis. Now, the third type, which is the focus for today, is chromatography. I guess another way of looking at what analytical techniques actually means, a very simple way of looking at it, as I've mentioned in this PowerPoint here, is it's really about finding what's in stuff. Very technical term, I know, using the word stuff, but really what we're saying is what is in various substances. And we have chemists have a number of types of techniques they can use to do that and to analyse substances and work out what's actually in them. So let's look at chromatography. The key, just moving to the next slide, sorry. I guess one of the key concepts you need to understand to grasp the principles of chromatography is that of secondary interactions. Now, we covered that earlier in the year. You also covered it quite extensively in stage 1. In particular, just essentially the differences between a polar bond and a non-polar bond, or more importantly, a polar molecule and a non-polar molecule. So you may recall the polarity is all around charges on molecules. Some have high polarity, meaning there's a significant charge on that molecule. Others have very little charge or virtually no charge, and they are known as non-polar molecules. So we have the two types here, non-polar and polar. The other principle that's important is around that polar molecules are attracted to other polar molecules because of the charges that are on there. So if you have two charged particles, then they're going to be attracted to each other. Similarly, and probably a little more complex, non-polar molecules are attracted to other non-polar molecules. So it's that attraction between a non-polar molecule and another non-polar molecule and a polar molecule and another polar molecule that is really one of the key principles behind how chromatography works. So we just need to keep that in mind as we're looking at this particular technique. So, kind of slide I've already really mentioned, already talked about. So, yeah, polar substances are attracted to other polar substances. Non-polar substances are attracted to other non-polar substances. Okay. So, chromatography essentially is an analytical technique that utilises differing polarity of substances to separate the components of mixtures. So we can get a mixture that would have may have a variety of substances within it, different components of that mixture, and we can separate them and actually then identify the different components based on their differing polarities and how they are attracted to different things because their polarities vary. I'd like to understand what these words mean very shortly, but I'll just mention to you, or just start off by saying that in chromatography we have two parts or two components. One is a stationary phase, and the other is a mobile phase. And these are actually physical components of chromatography. The stationary phase is usually a solid material, which components of a mixture will interact with on its surface. So it's a solid substance that different components of a mixture will interact with at different levels. This interaction occurs as adsorption to its surface. Now, I'll talk a little bit about what this word means here. You've probably heard the word absorption before, but adsorption is actually a different phenomenon. So we'll look at that shortly. The mobile phase is the other part of a chromatography setup, which is a fluid substance, which means it's either a liquid or a gas, that moves across the stationary phase, carrying the mixture to be separated. So the mobile phase will carry the mixture that we're trying to analyse and trying to work out what might be in it. Okay, so that might give you a bit of an idea what chromatography is, but probably you're still a little bit unsure how it works and what it looks like. So essentially, each of these, we have the stationary phase and the mobile phase, and they will have different polarities. The stationary phase might have a greater polarity than the mobile phase, and or the opposite way around, could be the mobile phase has a greater polarity than the stationary phase. Okay, so as a mixture moves through the chromatography system, the relative polarity of the different components will result in them being more attracted to one of the phases. So let's say we've got a polar stationary phase and we've got a non-polar or a much lower in polarity mobile phase. If we had a polar molecule, that's going to be much more attracted to the stationary phase because it is also polar. Whereas if we had a non-polar molecule, it would be much more attracted to the mobile phase, which is also non-polar. So it really depends on the relative polarities of the mobile and stationary phase, which one the substances within a mixture will be more attracted to. As I've just said, this results in the various components of a mixture separating out and adsorbing at different points along the stationary phase. Now, I've said this word adsorption a number of times, which is probably confusing you because it sounds way too much like absorption, but it's actually a slightly different thing. We'll start with what absorption means, which is something you're probably familiar with. It's the incorporation of particles into the body of another material. So essentially, we all know if you absorb something, something gets absorbed, it is incorporated into the body or into the main part of that substance. Whereas adsorption is about the attachment of molecules to the surface of a solid. Okay, so adsorption really is just things attaching to the surface. So you can see that's illustrated here in this diagram, that adsorption is when it's attached to the surface, as we can see here on material A. Material B is absorption, where the materials are actually incorporated within the actual substance. In chromatography, we are dealing with adsorption, so you can kind of, for the time being, forget about absorption because we won't really be talking much about that. Okay, so just to recap what I said earlier, and just to sort of give a step-by-step sort of description of how chromatography works. Normal phase chromatography, which is the most common type of chromatography, the stationary phase is more polar than the mobile phase. So simplistically, we would say the stationary phase is polar, the mobile phase is non-polar in most types of chromatography, and that's what we call normal phase chromatography. There is some less common types of chromatography that use reverse phase chromatography. That is where the stationary phase is less polar than the mobile phase. Okay, but usually this is the case. So these are the steps within chromatography. Okay, the mixture that is to be separated is introduced to the stationary phase. We'll show a little diagram in a minute that will give you maybe a bit more of an idea of how what it actually looks like. But we have a stationary phase. Remember, it's a solid substance that the mixture is introduced to. Okay. Then the mobile phase is allowed to flow over the stationary phase. So we have a solution or it could, in some cases, it is actually a gas that is the mobile phase. Most cases, it's a liquid, all right, that will flow across the stationary phase. When this mobile phase does flow across the stationary phase, or through, and in fact through the stationary phase, the components of the mixture will adsorb to the stationary phase or stay within the mobile phase to varying degrees according to their level of polarity. So probably another point that I probably should have put here is that when the mobile phase moves along the stationary phase, it brings the mixture with it. Okay, so it has to be something that the mobile phase has to be something that will take the mixture with it as it goes along the stationary phase. And really then it's all about whether the different components of the mixture will attach or adsorb, be more attracted to, is another way of looking at it, to either the stationary phase or the mobile phase, depending on their level of polarity. Okay. Just explain this type of chromatography, which is the most common, and the next slide has a diagram of it, which gives you a bit more of an idea of what it looks like and how it works. So the most common type of chromatography is what's called thin layer chromatography, okay, where we have a thin layer of silica or alumina, and that is coated onto either glass or a plastic plate. Okay, it's actually like a little strip of glass or plastic, and we have silica or alumina on it. And silica and or alumina are polar. Okay, that is a polar stationary phase. So like the first type that we mentioned before, this is normal phase chromatography. Okay, so once the mixture has been applied, it is placed into a container containing a shallow amount of non-polar solvent. Okay, now this non-polar solvent is, in fact, the mobile phase. Okay, it is the solvent, the liquid that will be carrying the mixture with it across the stationary phase. Okay, so the easiest way to get an understanding of this is to look at a diagram of it. So thin layer chromatography would involve this component here, which is the stationary phase. This is the thin plate that is coated with either silica or alumina, and it is polar. The liquid within this tank, which is down here, I should say, the liquid that is within this tank, all right, is our mobile phase. Okay, that's the solvent. So when the plate is placed into this liquid, the solvent or the mobile phase will flow, and that is just through what's called capillary action, which it's just really just the liquid diffusing across the plate. So the liquid will move through the stationary phase, and it will carry the mixture with it. Now, in this diagram, the mixture of these little dots here. So there's three samples, I guess, of the mixture here. 1, 2, 3. So essentially, just three little drops, I guess, of the mixture has been put on this plate. The mobile phase will carry that mixture with it. Okay. Now, dependent upon how polar the different components of this mixture is on these three different samples, will determine where each component will attach and separate out of the mixture. Okay, so if we look at the after diagram here, we can see that there's one component, that's these three blue dots here, that's separated and adsorbed to the stationary phase here. We can see another component, which is red, that has adsorbed to the stationary phase up here, and the final component of this mixture is separated out and adsorbed up here. Okay, so that would tell us that this substance, okay, in the mixture is much more polar, or is more polar than this substance, because it was more greatly attracted to the stationary phase than this one, and therefore adsorbed and separated out earlier than this one. And finally, the yellow one would be considered the least polar. Remember that our stationary phase is polar, so the earlier it separates out and adsorbs tells us whether it's how polar it is. But this one would be the least polar because it took the longest to absorb two